Hi guys, welcome back. This is stage 2 signal transduction. So we're looking at the 3 stages of cell communication. You don't want to start with this video. Things won't make sense. Go all the way back to the overview video, or at least back to receptor activation, and work your way through the 3 stages in order. But what we want to do is we want to look at some of the primary ways that the signal can be relayed from the cell surface receptor to something inside the cell that can actually respond. How do we get away from that cell surface receptor to the response proteins? We call this signal transduction. Now there are 3 main ways that the signal can be transduced inside from the cell membrane to some sort of cell response proteins. In this video we'll take a look at what's called a phosphorylation cascade, and then something similar but slightly different that's typically found in bacteria, fungi, and plants, called simply a 2-component regulatory system. And then I'm going to save second messengers for another video because they're a little bit more complex. So let's start with a phosphorylation cascade. So we've got a signal of some kind. We've been seeing that in earlier videos. It binds to the receptor and activates it. The active form of the receptor is a protein kinase, meaning that it's going to take an ATP, it's going to hydrolyze that ATP, releasing both energy and phosphate, and use it to phosphorylate a soluble protein, one that's not bound to the membrane, so it can then diffuse within the cell. We'll just call it protein kinase B, and the receptor could be protein kinase A, for example. And then we simply get a series of phosphorylation reactions where kinase B phosphorylates the next one, which phosphorylates the next one, which then phosphorylates the next one. So each phosphorylation reaction activates the following protein kinase. In the end, some sort of cell response protein needs to get activated as well, and we'll talk about what those different cell response proteins might be. This is what we call a phosphorylation cascade. So let's take a look at one as an example. This is epidermal growth factor. Epidermal growth factor is what's called a mitogen. It stimulates mitosis in epidermal cells. It's a hormone, so here's the EGF hormones out here. Here's the cell membrane at the surface, and there are EGF receptors. When an EGF hormone binds to an EGF receptor, if it's just a single receptor, nothing happens. But when a nearby receptor also binds to EGF, that draws the 2 of them together to form a dimer. And then the first thing this dimer does is it autophosphorylates. In other words, it phosphorylates itself. Once it's in this dimer state, autophosphorylated, it's then going to be able to interact with, and remember each of these changes has to do with shape changes due to binding of the proteins. It's going to be able to interact with a series of what are called relay proteins, which are peripheral proteins. So they're not completely transmembrane, but they're not soluble either. First one is called GRB. When this dimer has been autophosphorylated and interacts with GRB, that forces GRB to interact with SOS, which then forces SOS to interact with RAS, R-A-S. RAS, when it's attached to the membrane, is carrying a GDP, right? The sort of spent version of GTP. But when SOS binds to RAS, that forces RAS to let go. It changes its shape so it no longer is affiliated with the membrane, becomes soluble, and once it's soluble, that forces it to let go of the GDP and replace it with a GTP. Once RAS is soluble carrying GTP, it has a strong attraction for another protein called RAF, and that combination has a strong attraction for a protein called MEK. This 3-protein combination carrying GTP then acts as a protein kinase to phosphorylate the MEK protein. When MEK gets phosphorylated, MEK and RAF stay together, but they let go of RAS. RAS then is carrying a GDP, and it doesn't like being soluble with GDP, so it snaps back to the membrane, and it's available for another round of signaling. Meanwhile, this RAF-MEK dimer, heterodimer, 2 different proteins dimerized together, phosphorylated, act as a kinase to phosphorylate a protein called ERK. Phosphorylated ERK is now a protein kinase that migrates just by diffusion into the nucleus, where it phosphorylates 2 more proteins called MYC and PHOS. And there's actually a 3rd one called Jun that's not in this image. But MYC, PHOS, and Jun, when they're phosphorylated, actually control the transcription of key genes involved in mitosis. So we go from some other cell somewhere in the body producing a hormone called EGF to the cell ultimately responding by transcribing and translating genes that are important, critical to mitosis as a way to trigger and initiate mitosis. Now, we said that EGF is considered a mitogen because it generates or stimulates mitosis. The kinases involved in a phosphorylation cascade that trigger mitosis, such as this example here, are called MAP kinases, for mitogen activated protein kinases. Why do I mention these? Because these are a major focus, not the, but a major focus of cancer research. If something goes wrong, either with the mitogens themselves or with the MAP kinases, we often have a situation leading to cancer, cell growth that's out of control. These are all critical steps to maintain control over cellular growth. So take a look at this, probably hit pause or rewind, watch this again, and think through all the steps. You do not, my class, have to memorize the names of all of these proteins. You should remember EGF, EGF receptors, and maybe the response proteins, McPhos and Jun. That'll get you where you're going. And remember what the response is. Remember what a MAP kinase is, what a mitogen is, and what EGF stands for. But these kinases in this system, then, these MAP kinases, are really important in understanding cancer. Okay, the second one I wanted to talk about in this video is not a phosphorylation cascade, but involves phosphorylation. It's a much shorter, more direct route, and no surprise, it's really common in bacteria who tend to do things in a very short, efficient, direct manner. This is what's called a two-component regulatory system. In a two-component regulatory system, the bacteria receive the signal from the environment by binding to the receptor, and the receptor is an enzyme-linked receptor, referred to as the sensor kinase. In this case, the signal is nitrate or nitrite. These are inorganic forms of nitrogen that the cell can bring in for its various metabolic needs. Right? We need nitrogen. All living things need nitrogen in their diet, so do bacteria. NARQ is the protein that's referred to as the sensor kinase. And just like when we talked about enzyme-linked receptors, when nitrate or nitrite binds in the extracellular environment to NARQ, it changes its shape and activates its protein kinase activity. And it's interesting because the first thing it does is it autophosphorylates. And the autophosphorylation reaction then triggers transfer of that phosphate group to a target protein. And that target protein is called the response regulator, NARL. Now, whereas before in the last slide we were seeing the protein that was the target of the phosphorylation being yet another kinase, that's not the case here. In this case, this is the actual response protein. So NARQ is the sensor kinase, the receptor. NARL is the response regulator. In this case, the activated receptor directly phosphorylates the response protein, which, just like we saw with McPhos and Jun in the earlier example, it influences the expression of genes. In this case, genes necessary for the uptake and metabolism of nitrate and nitrite. Now, if you watched the earlier videos, particularly video number 1, the overview, you saw that this particular form of cell communication is simply referred to as a cellular response or cellular adaptation, because the signal, nitrate or nitrite, is not coming from another cell. It's coming from simply conditions, chemical conditions in the environment. All right, I'm going to stop this video here so you can absorb all of this, watch it multiple times, whatever you need to do. And I'm going to create a next video that will just focus on the third method of signal transduction, which is second messengers. I hope to see you there.